Welcome to today's lesson on the stages of genetic engineering, where we will explore the key processes and tools involved in manipulating the genetic material of organisms to achieve desired traits. In today's lesson, we will cover the main stages of genetic engineering, focusing on the use of restriction enzymes, ligase, sticky ends, and vectors. Each of these components plays a crucial role in how scientists can modify the DNA of an organism effectively. So far, you have learned about what genetic engineering is, the direct manipulation of an organism's genes using biotechnology. This foundational understanding of genetic engineering sets the stage for us to dive deeper into the specific tools and steps involved. This knowledge is pivotal because understanding how each component, restriction enzymes, ligase, sticky ends, and vectors, functions allows us to comprehend how genes can be precisely cut, pasted, and transferred. This paves the way for advancements in medicine, agriculture, and many other fields. Let's get started. Today, we'll look at the fascinating process of genetic engineering in more detail, a powerful tool in modern biology. This process allows scientists to modify an organism's genetic material to achieve specific outcomes, such as producing a desired protein. We'll break down this process into clear stages, highlighting the roles of restriction enzymes, ligase, sticky ends, and vectors. The first step in genetic engineering is to identify and locate the gene of interest in the original organism. This gene is responsible for the desired trait or protein. For example, if we want to produce human insulin, we would isolate the insulin gene from human DNA. Next, restriction enzymes come into play. These special enzymes act like molecular scissors, recognizing and cutting the DNA at specific sequences, precise locations. When the restriction enzyme cuts the DNA, it leaves behind sticky ends, which are short sections of unpaired DNA bases. These ends can easily bond with complementary DNA sequences. Next, we take a bacterial plasmid, which is a small circular piece of DNA found in bacterial cells. The plasmid is used as a vector to carry the isolated gene into the bacterial cell. It is also cut using the same restriction enzyme. This ensures that the sticky ends of the plasmid match those of the isolated gene. Once both the gene and the plasmid have matching sticky ends, they can be joined together. This is where DNA ligase plays a crucial role. This enzyme works like glue, joining the sticky ends of the gene and the plasmid together. This forms a single, continuous DNA molecule, known as a recombinant plasmid. The newly engineered plasmid is then inserted into a bacterial cell. This process is facilitated through various methods, such as heat shock or electroporation, which make the bacterial cell membrane more permeable to the plasmid. Once inside the bacterial cell, the plasmid begins to replicate as the bacteria reproduce. Because plasmids can multiply independently of the bacterial chromosome, each new bacterial cell will carry copies of the recombinant plasmid. Finally, the genetically engineered bacteria are placed in a fermenter where they reproduce rapidly under controlled conditions. As they grow and divide, the bacteria express the inserted gene and produce the desired human protein. This can be harvested and purified for various uses, such as medical treatments or research. Let's work through some questions to consolidate our knowledge. Our first question is, how do restriction enzymes aid in the process of genetic engineering? Pause here to have a go. Now let's go through the answer. Restriction enzymes act like molecular scissors to cut DNA at specific sequences. The DNA is cut into fragments, creating sticky ends that are crucial for the next steps. Let's look at a second question. What role does the enzyme ligase play in genetic engineering? Pause here to have a go. Now let's go through the answer. We have learnt that ligase is used to join pieces of DNA together. Note how the sticky ends of the DNA fragments are complementary and can pair up. Ligase facilitates the formation of bonds between the sticky ends, effectively sealing the DNA fragments together. Let's look at one last question. Why are vectors important in genetic engineering, and how do they function? Pause here to have a go. Now let's go through the answer. Vectors are vehicles used to transfer genetic material into a host cell. The vector, often a plasmid, has the new DNA inserted into it. Next, the plasmid vector incorporates the new gene, often using restriction enzymes and ligase to insert it. Then, the recombinant plasmid is then introduced into a host cell, which will replicate the new genetic material. In today's lesson, we explored genetic engineering, focusing on its main stages and tools. We learned that genetic engineering involves altering an organism's DNA to achieve specific traits. We discussed how restriction enzymes, acting as molecular scissors, cut DNA at specific sequences to isolate the gene of interest. Sticky ends, or overhanging nucleotide sequences, help the gene of interest bind to the vector DNA. Ligase, the molecular glue, then joins these DNA fragments into a plasmid or vector, forming a stable strand. Lastly, we covered vectors, such as plasmids and viruses, which transport the gene into the host organism. The genetically engineered bacteria is then used to produce the desired protein. Understanding these components, restriction enzymes, ligase, sticky ends, and vectors, clarifies how genetic material is precisely modified and inserted, with applications in medicine, agriculture, and research.